Internet tools and applications. These computer programs, actually tools that are found in gene banks, in databases, in different genetic websites that have been established by different organizations, different countries. Just like what I've said before in introduction to database. So, but in this lesson, I will be, you know, showing you the exploration of what these tools are all about and how we can, have your way to search for tools, to search for certain things, search for, data, search for protein sequences, DNA sequences, RNA sequences. They are open to everybody. You can search anything you want depending on whatever you are working on. So that's why these tools are, and there are certain things that you can actually do, with these tools to, in terms of genetic analysis, you know. So, but we're going to explore all of them so that you will be able to see very well. So just follow me very carefully. Thank you. So we have terminologies. So there are certain terms in bioinformatics when it comes to the aspect of internet tools that you need to be aware about. You need to be familiar with. You need to understand. There are language. For every term, for every field, there are always language that are associated with them. So you need to understand the language of internet tools to be able to know what you are doing. So now we'll be looking into these terminologies one after the other. Now we have gene banks. What are gene banks? So just so we have gene banks. What are gene banks? The gene bank sequence database is an open the gene bank sequence database is an open access, means is open to everybody, to you, to me, to everybody. You can search whatever you want. You can search for data, any kind of data you want. You can search for data that are associated to plants, animals, microorganisms, maybe data of a particular genes, mutated genes, cancer genes, breast cancer, any kinds of genes. In as much as these genes are associated, in as much as you are working on a living organism, you are working on a living things, living organisms. You are working on genetics. You are working on molecular biology. You are working on molecular. You are working on molecular data. So we have annotated collection of all publicly available nucleotide sequence. Now, if you mean annotated, we mean hidden informations that have been analyzed by certain individuals and allow access by anyone on the database on gene banks. So, and then we have protein translations too. We have sequences that have been translated, so you can actually access those sequences and maybe do whatever analysis or research you want to do on those particular sequences. This database is produced and maintained by the National Center of Particular Information, which is NCBI, as part of the National Nucleotide Sequence Database Collaboration. So we have BLAST. Now this BLAST is actually the main and primary tool when it comes to internet tools in bioinformatics. And the essence of this BLAST, which is the essence of this BLAST, is an algorithm for comparing primary biological sequence information. And this BLAST is an algorithm for comparing primary biological sequence information, such as the amino acid sequence of proteins or the nucleotide of DNA or RNA sequences. So what this means is that BLAST tools is used to search for similarities and differences between two or more nucleotide sequences, or be it protein sequences. So that's the essence of BLAST. So you can search for similarities, you can search for differences, maybe mutation regions and things like that. So that's the essence of BLAST. You can also use BLAST to work on evolutionary relationship and so many things like that. But we're going to be talking about BLAST. We'll explore it, we'll do some certain practicals for you to understand what really BLAST is all about. So, and we have a query sequence. Now query sequence is the original sequence you have. So for instance, you have a particular sequence in your system and you don't know the name, you don't know what organism it belongs to, so you can actually use that sequence by blasting. So that means you use a blast too. So, but the original sequence you have, which you don't know anything about, is actually the query sequence. So we have our accession number. Our accession number is a unique identifier given to a DNA or protein sequence record to allow for tracking of different versions of that sequence record and the associated sequence over time in this single data repository. Now, what this means is that accession number is actually a unique number that have identified to a particular sequence on the database. So as soon as maybe you submit a particular sequence on the database, so the database will assign a unique number to that sequence, and that unique number is called accession number. So you can actually use this accession number to to search for that particular sequence on the gene bank or on the database. So we have its value. The blast score indicates the quality of the base alignment between the query sequence and the found sequence. So the its value, which is the blast score, indicates the quality of the base alignment between the query sequence. So you will understand that. So as we go, the expect value is a parameter that describes the number of each one can expect to see by chance when searching a database of a particular size. So you will also understand what this expected value is when we begin to do the practical applications. So we have the practical applications now. We have search for data, we have download of data, which is single and multiple, we have submission, we have blast. Now this is actually giving you a brief introduction of what the practical applications will all be actually. Now the first thing we begin to explore is search for data when it comes to internals, theories and other things. So what do we understand by search for data? Now we use search for data to actually look for certain data that are found in the database. So how can you search for data? That's what this is all about. How to search for data, and how you can retrieve data. Maybe you're actually working on maybe a research on mutation. You're working on a research on maybe on a microorganism. Maybe that particular microorganism have a hidden future that solve a particular problem, that is solve it cures a particular issue. So all you need to do is maybe, or you want to search for that particular data, you can just do a lot of things. We'll explore that as time goes. Then we have download of data. So after you search for data, the next thing you do is you download the data. And we have submission. So maybe you have after maybe you have walked over the sequence, you have analyzed, you have made analysis, you have made discovery. So you can actually submit that sequence, that nucleotide sequence, RNA sequence, or protein sequence on the same database, on GenBank. You can actually submit them there, so that any other person elsewhere can actually see into what you are doing. Now we have blasting. So we would talk about blasting as we keep going. So we have search for data. Now nucleotide sequence search. So we have nucleotide sequence of different and diverse plants and animal, which can be searched on available gene banks and be downloaded for further studies and research purposes. So that is pertain, specific for nucleotide sequence search. Now you see, this nucleotide sequence search is actually specific to only DNA or RNA. So what we have diverse and in different genetic data file format, just like we have PDF, we have Word, things like that, Excel. We also have data file format for every sequence data in the database. So we have FASTA format, we have FASTQ format, we have BAM, we have SAM, we have CSV, GTF, and a lot of them. So as you can see on the screen. So this is an assignment for you. Please get and read about the full names of the above genetic data file. This is your first assignment. So you will be submitting this assignment to me on my email. So note it down. You can write it down. So Let's keep going. And we have, okay, notes. You might be provided with a accession number or a sequence ID for retrieval from a gene bank, or be given the name of the sequences you are to search for and information about it. Anything at all is of importance value to accomplish your mission. So gives us a note, a reference, a scenario, or we'll work on it as we go. So there are three major ways to search and download data sets from database. Now we're actually talking about searching of data on databases. So number one, we have retrieval method. Number two, we have direct searching, and number three, we have blast, the use of nucleotide sequence or protein sequence. So now let's move on. Retrieval method. What does it mean? When it comes to maybe you're searching for data, we have retrieval method to be number one. But what is retrieval? What does it mean to retrieve? Now this is done when you have an accession number. You cannot retrieve until this or what you want to retrieve is already on the database. So what you need is an accession number. So just as I've said, it's a unique number given to a DNA protein sequence recorded to allow for tracking of different functions of that sequence record and associated with sequence over time in single data repository. So that's a accession number, just like what I've explained before. And this method can also be done to search for literatures and research materials. Maybe you want to search for a particular research materials, you're doing your project work, maybe you want to search on a particular microorganism, you want to search on a particular plant, you want to start on a particular drug, whatever that is associated with genetic, with drug, with health, with plants, with agriculture, you can actually search for it. Just like Google, you can actually search for it on NCBI database, just like what is here, this place, NCBI, the particular database. Don't worry, you will understand how those databases look like when we begin to deal with the practical aspect of it. So we have direct searching. Direct searching is also a method used in the aspect of searching for data. Now this is best done when you have known the organism name, the disease name, gene name, or that of any nucleotide sequence, either from plant or animal, or even microorganisms. But what you need to understand is that you need to know the name of what you are searching for. You need to be precise. Hence, the name is imputed into the search space. Advanced search can also be done in this manner with precise target. So I'm going to teach you what all that is all about. I mean the practical application. So just be ready. So the other one, we have blast. Just like I've said, you have a particular sequence, you don't know the name, you're just there. We're going to work on that, don't worry. So all you need to do is blast. So as you blast, it will bring the name of that sequence in particular, what that sequence is in particular, where the organism the sequence actually belongs to. So blast will show you all that, the total number of the nucleotide bases in that sequence. You will know. So now the use of nucleotide sequence, which is blast, which means in full basic local alignment search tool, just like I say, internet tool. So is an internet tool. So blast is an algorithm for comparing primary biological sequence information to know about the differences, the similarities between those two sequences or more than two sequences. You want to know the differences they have in the nucleotide bases. Maybe this particular nucleotide base has maybe a 500 nucleotide bases in the sequence, and the other one has 600. So blast will tell you that this is 500, the other one is 600. Maybe you want to look at similarities and differences. So blast will tell you that these two sequences or more than two sequences have what we talk about maybe let me say 100% similarities, resemblance. Maybe it's 60, maybe it's 50. So blast will tell you. Now the basic element search to find regions of local similarity between proteins or nuclear sequence, just like I've said, so and things like that. Blast can be done on different databases. So on different databases we have types of blasting. We have nucleotide blast. Now the essence of this nucleotide blast means you are blasting a nucleotide sequence against a nucleotide the gene bank. So we have a protein blast. So you are blasting a protein sequence against a protein, you know, database or gene bank. So we have nucleotide 6' translation proteins. So we have large number of query sequences. They are all there. We're going to be dealing with that. So just follow me carefully. We have databases for BLAST. We have NCPI databases. We have ANCBIL. We have KEG. We have Interpro, and others, which I've already explained in the previous class. So in the next class, we shall be exploring the practical applications one after the other. So be ready, and be focused, be